to welcome everyone to this month's version of the ReproNim webinar. We're super excited to have an old friend and colleague, Anisha Keshavan, here to speak to us. Anisha got her BS in aerospace engineering and applied mathematics at the UCLA. She did, I think the web told me this, so I hope it's true, a dual PhD, which I don't see all that often,
Oh,
in
just
biomedical
one.
engineering. Just one?
One PhD,
Okay.
well, schools.
Two schools, dual school, one PhD, two schools?
Yeah, one.
Okay.
Yeah.
Okay, well, we'll blame Octave for the way they put it on the website. But anyway, a PhD in cool things in biomedical engineering and biomedical engineering, bioengineering and biomedical engineering at UC Berkeley and UCSF. Currently a senior data scientist at Octave Bioscience, following work that many of us in the ReproNim team have run into during her postdoctoral fellowship at University of Washington and her time as a research scientist at the Child Mind Institute. So without further ado, I will turn the floor over to Anisha.
Cool. Hello, everyone, future YouTube viewers. Little about me, which Dave already covered. I got my PhD at two different schools, Berkeley and UCSF, but just one PhD in bioengineering. And my focus was in multiple sclerosis neuroimaging. And then I did a postdoc at eScience Institute, which is really a data science institute. And I've been at Octave now for 4 years, but my biggest and most difficult accomplishment are my 2 children. So I always like to point them out because, wow, way harder than I expected. Okay. So what is Octave? We're a company for a precision care solution for multiple sclerosis. So why multiple sclerosis? Well, it's a really expensive disease. For almost $5 million in a person's lifetime. And then on top of that, there's kind of the usual comorbidities that, a lot of Americans have that increase the disease burden and the disability and the cost. And there's a lot of variation in outcomes where half of the patients are in disability in 12 years, but it's hard to predict early on who those people are going to be. And there's many, many drugs. I mean, already 22 plus drugs and more coming out every year. They're pretty effective, but they also cost a lot of money. And in some people, they are not effective. So it would be very unfortunate to go on one of these very expensive drugs only to find out it doesn't work for you. So I think everyone kind of needs help in this space. Patients, they have limited support, lack of clarity about their disease. Physicians don't always have all the information they need to feel like they're making good clinical decisions. Healthcare plans are like, wow, MS patients cost a lot of money. If they are healthier, then, we wouldn't be paying for unnecessary ER visits. Everyone wins if we can avoid the ER. And then, the life sciences in general, we need better tools to measure the biology of these patients. And so our company really consists of 3 parts. There's a biology team. We have a wet lab, and they're measuring proteins in the blood that can tell us more about the disease. There's, of course, the imaging part where I come in. And then we have a clinical insights team where we have a nurse that checks in with our patients every 2 weeks and triages them so we can try to avoid kind of these costly ER visits and all that kind of stuff. So I want to focus, though, on the imaging in MS here because this is the ReproNim talk. So I want to talk about the current state of radiology reports. And this isn't a real example. It is pretty rare, but it is dramatic. So I'm going to show it here. In between a wad of text, there's the part that the neurologists will want to pull out, and it's the impression. And even when they pull it out, it says mild, non-specific white matter changes unusual in a patient of this age. That's it. So it's not MS specific. It's very vague. It's not great. And so what would the ideal MRI report look like for neurologists to feel like they can make a decision based off an imaging report? Well, it would be MS specific. It would have that impression instead of buried in the middle of all this text. It would be at the top, like, these people are busy. And ideally, it would contain quantitative information. So the number of lesions an MS patient has by the region, and whether these lesions are new, are they stable, are they enlarging or shrinking? We know in the research world that lesion volume and change is also relevant. and of course, brain atrophy and regional atrophy. If we could nail this information and get it right, it could make a huge difference in these patients' lives and make a huge difference for the neurologists who are treating these patients. And then this kind of seems sad in the year 2023, but a lot of neurologists don't have access to just like the raw images of their patients, just because like of, I don't know, old computer systems. I don't know. You would think this part is pretty simple, but it's not. So Octave, we're trying to, or at least the imaging team, we're trying to address all of these needs. But for the neuroimagers out there, you know that this part, the devil is in the details, right? It sounds simple. Yes, count the lesions, measure the brain volume and the atrophy. But we all know this is very hard. It's hard to get right and it's hard to get reproducible. But it's also very important. So you can imagine that if you're a patient or, you know, treating a patient with MS and they're on a drug and suddenly you see that they have new lesions or lesions that are growing, or maybe you're seeing that their brain is shrinking at a abnormal rate, you know, abnormally high rate, you would think, well, maybe the current drug that they're taking isn't effective and it's time to start, you know, putting them on a more potentially more effective drug, but that has more side effects. Maybe it is now worth the risk if you had this information and it was reliable. So it's extremely important to get right, but also very difficult. And here, of course, reproducibility is critical because if we get this wrong, you could change the course of treatment for a patient, you know, on something that's not reproducible. That's none of us want to be in that situation. So reproducibility is important for science as a whole, but it's also very important if we want to bring that science into the, you know, clinical sphere. So what do we do at Octave to address this? So I want to break down why this is specifically hard with real world data. So there's a lot of human variability when a neuroradiologist looks at an image. And then there's, of course, the data quality issues we all know and love. There are different MRI scanners at various imaging centers. They all use different sequences year over year. So if you wanted to compare MRI scan from a patient at baseline with, you know, 2-3 years later, they've gone on a different scanner. It's not comparable anymore. What can you do? There's also the fact that clinical imaging centers are very different from, you know, our research where we're very careful. Maybe we put people in these like head coils and we pat it so they don't move. Clinical imaging centers just need to get images that are good quality enough for a radiologist to make a call. They don't necessarily care to take the time to make sure the image is, you know, able to be segmented by something like FreeSurfer. And so this is a problem. And then, of course, because of this, there are differences in our automated segmentation algorithms as a result. And so this then leads into the challenge to scale, because we know that no algorithm is perfect. We always need humans to check the output before we send out a report that says this patient has 3 new lesions. We need to make sure they actually do, in fact, have three new lesions, even though maybe their sequence changed and the scanner changed compared to the previous year. So I want to break this down. So differences in neuroradiologist impressions. So we had a little study where we had 90 different MS patients with each with 2 scans that were sometimes on different scanners and sequences. And we asked 3 neuroradiologists to rate them on a variety of things. So, you know, count the number of enhancing lesions. So these are GAD enhancing lesions. They were very good at this because that's the normal baseline job. If you're a neuroradiologist, you're going to go and you're going to count the enhancing lesions. So we were like, okay, great. They agree on that. But then you get down to the presence of enlarging lesions. This is a lot more subtle, and they did not agree on this. Brain atrophy, which we know in the research world is very important, especially for the progressive form of the disease. When we asked for their impressions on, you know, how severe this atrophy was, you know, from like not severe to, you know, medium to severe, they didn't agree. So this is our baseline. And so what we also did was we ran these images through an automated segmentation software called Cortex, which is an offshoot of FreeSurfer in company form. And we found that if we kind of helped them by, you know, coloring in existing lesions, they were able to detect a lot more new lesions. So now the question is, does this software help them become more reproducible, right? Are they more self-consistent and are they more consistent with each other when they have the aid of automated segmentation tools? So that is an open question that we're currently in progress studying. Okay, So data quality issues, the MRI scanner sequence differences year over year. So just how bad is this? So we went to one of the leading, MS centers, just a single MS center. So this is a neurologist group that focuses on MS, but they don't have a scanner. They have to send their patients out to get their imaging scans. And so here we found that 58% of patients that received longitudinal scans, they varied across facility, field strength, manufacturer, model, and serial number. So even if you go to the same neurology clinic, you're going to go to a different scanner sequence. You're going to get some sort of different image the next year. And so that can make comparison to the previous year's MRIs really difficult. So these are some problems that I think are structurally harder to solve because you have to kind of change people's way of doing things normally. So moving on, clinical imaging centers need to scan lots of people quickly. And so the scan quality itself can really vary. So how much does it vary? Well, we did a little study where we ran MRI QC on all of our images, and we tried to group together people that had scans that were very similar to each other versus very far apart from each other. And so here are two extremes where in this set of T1 and FLAIR images, they're very close to each other in terms of their noise, just visually. Whereas you get something like this, where this initial FLAIR sequence is pretty low quality, and then this one is quite a bit different. So this is what we're dealing with. So what does that mean for our estimates of atrophy? Well, now it's a little scary to do. And so how do we approach this? So what we're trying to do right now is to model this statistically. And so what we've done is we've created this GAMLS model. So GAMLS is what the World Health Organization uses to create these like growth charts for kids, for example. So we took that and we modified it such that we can add the T1 CNR metric from MRI QC. And we found that the model fit a lot better. So now we can have these, I want to say atrophy charts, not growth charts, atrophy charts for MS patients, and where we can kind of get an idea for the context of a certain measurement, given the fact that maybe the quality was not as good as it could have been. So next, we're thinking about longitudinal changes. So this one is interesting. So if you're an MS patient, you probably have a pretty low brain volume. So if you were to take your brain volume and compare it to a normative distribution of healthy people at your same age, it would probably be pretty low. And there's not a lot of information you can get year after year. It's like, okay, you have low brain volume percentile, and then the next year, yes, you've maybe fallen to 2 percentile points. Is that a lot? Is that a little? And so what we've done here is we're trying to give context to the next time point conditioned on the previous time point. So in this chart over here, we're seeing kind of 2 centiles. One is this cross-sectional centile. So an MS patient at the age of 37 was above the median in terms of their overall, single cross-sectional time point. And then 3 years later, they're still above median. So to a neurologist, this would be like, oh, okay, I don't know how much information I can get out of this. But actually, if you condition on this initial measurement, you find that their total brain volume is actually pretty low. This drop in brain volume was not what we would have expected for a patient of this age with this scanner change and with this contrast to noise ratio. So here's some more information about various ways we're taking just right now visually looking at these scans and comparing the quality over time and visually, checking to see, did their ventricles actually grow? Did their brain actually shrink? And how does that correspond to these various centiles? And so the study we have in progress now is, can this statistical modeling of normative brain atrophy that also includes image heterogeneity lead to improved clinical interpretation of brain atrophy in terms of reproducibility and clinical impact? So we're trying to take all these years of research on brain atrophy and give it meaning to a clinician in the clinic. So very excited about this project. Okay, and so then finally, this challenge to scale. This is a problem I have been working on for a very long time, and it's still very hard. No algorithm is perfect, and humans need to check the output before sending out the report, right? We don't want to tell a patient they have a new lesion, which causes them to change their medication if they didn't actually have a new lesion. We also don't want to miss a new lesion. So how do we address this? So if you know my work from the past, you know, I'm a big believer in collaborative work and doing this on the web. And so at Octave, we have developed kind of this in-depth, fully featured lesion editor, which I'll show a little video of. And then our long-term goal is to kind of translate this to Brander style swiping. So I'll play this video. So in this example, we have run data through Cortex, which is, you know, a free surfer variant, but Cortex has missed this cerebellar lesion because it's kind of tricky to segment that area in the first place. So what our user here is doing is they've clicked the center of the lesion and they're looking at it in 3D and they're deciding, you know, the bounds of this kind of like level set method that we're using here. And so we have a lot of different ways of annotating this image. We work in conjunction with neuroradiologists. So after our first level of folks do an initial scan through, we then send this to neurorads who also use it to fix our edits. And so our lesion counts are as accurate as they can be with the human eye, the expert human eye. But the hope is, you know, this still can take, you know, on average 25, 30 minutes. And so we're hoping in the future we can kind of transition to a Brander type scenario where we kind of show a lesion and a user can swipe right or left if it is in fact a lesion or not. So that is our other exciting project at Octave, MRI team. And I think that's yeah. So what does it look like if we get all this right, if we can work through all these kind of details? What can we get? And so we've been working a lot on data visualization so that we can show clinicians, you know, who are maybe aren't used to seeing MRI scans, because oftentimes they don't even get to see the MRI scans. Show it what it looks like when we bring all this advanced neuroimaging features all together. So this is our 3D, you know, glass brain type model where, you know, you can see the subcortical structures here, you can see the folds. And let's see, I'll just wait for it to do its thing. The lesions will pop up at some point. And I think what's important to note here is that MS has a very distinct kind of pattern of these lesions. So it's good for neurologists to see that in this case, yes, the lesions are falling nicely along the ventricles as we would expect. We can see how those lesions are changing over time by morphing this volume because some lesions do in fact shrink, which, you know, indicates remyelination, which is a really good thing, but oftentimes they grow. And then let's see, we're gonna add some other stuff. How long is this video? Oh, tracts, white matter tracts, of course. Important to see where these lesions live on the white matter tracts. So we can kind of visualize that here. I'm gonna move forward, cortical thickness. And let's see. Then we kind of blend it all together with a kind of traditional view. And, you know, so we're just kind of playing around with this and seeing what really resonates with clinicians and hopefully eventually patients. All right. Right, so key takeaways, that kind of encompasses all of our work at Octave. So I think the key takeaway is that reproducible science can positively affect an individual if we do it right. But the neuroimaging in a clinical setting still poses a lot of challenges that I think we're still trying to address in the research realm, but also, you know, here in industry. And so I think what I also want to cover is just touch on the skill sets that you would need if you wanted to bring neuroimaging to an industry or clinical setting. And I think ReproNim really covers a lot of it. The reproducible basics module on version control systems and package managers. All of this is not optional at Octave. I mean, when we hire people, I want them to be able to write a Docker image and, you know, version control all their code and, you know some basic statistics. So I think what ReproNim is doing is very useful for transition to industry as well. So with that, I think I just want to thank all these people. The majority of the work was done by all these folks that you see here. Some of them might be familiar to some of you. And it's been a lovely time working with all these folks. So that's it. I will take questions now.
Cool, thank you so much. You emphasized human in the loop a lot.
Yeah.
And especially when it comes to telling a patient, you know, that yes, they have, you know, bigger or whatever lesions, I clearly see that. we get the sense that often in the research world, where we're not quite talking to any individual patient, we just have this big glob of results that we hope sort of statistically take care of any outliers. Do you have that same passion for the human in the loop when you're doing 10,000 subjects from the UK Biobank or some big data source?
Yeah, I mean, I think I feel like it's been a problem for a while. We've been collecting larger and larger data sets because we have all these questions to ask of our data, but the way that we process it now is so different than the way we did when we only had like 50 scans, right? And so, yeah, when you have 10,000, of course, the way that you do things changes, but then now our effects that we're measuring are maybe not as accurate as they should be. I don't know. I think it's a real challenge. And I think if we can figure out how to do it on the individual and really make it fast, and we can start applying it to these large scale data sets.
And ultimately, that was sort of a leading question that to some extent I would lean to not really being all that different. I mean, you may be talking to an individual about their individual result, but that could well be based on them relative to 100, 1,000, 10,000 other cases that
our reference data sets and building these longitudinal models they're all QC'd, but it takes a really long time.
Right.
Yeah. So
and one last variant of the same thing. In the old days, having been old enough to have done manual image segmentation, Andy and some others in the call know about that. And then the free surfers and things that come along and say, oh, get the humans out of there, they make mistakes, and here this automation will be perfect. And it's like, no, you look at it, automation's not perfect either. It's just it makes a different class of mistake. There are mistakes humans will make that the machine never will, and there's the mistake the machine makes that the humans never would. Why was I going down that way? But just to, I don't know, it's just more a reminder that even as the larger data sets get larger, the human, looking for the different types of errors, seem equally important, and that just has to be made more efficient, in order to really take advantage of these big data sets.
That's where visualization is super important. So on our Slack channel, I'm always taking pictures of like 2 time points and like focusing on the ventricles, and I'm asking my neuro-rads, do you think it's bigger or smaller? And I've been like testing them unofficially to just, if you don't see the difference, like actually see the difference, I don't know how comfortable you would be saying like, oh yeah, your atrophy is bad. It should be visual. So it's all, I think for me, it's always about making the right picture to just sanity check what the computer's doing, because I don't trust it. Yeah.
And I think that's a great point. The QA, QC, maybe not, looking at every voxel in one way. It may be how do we generate cooler visualizations
like that snapshot that
illustrate the types of things.
Yeah.
Cool. Okay, I've usurped the first set of questions. Others from the audience.
I'm going to piggyback. Hi, Anisha.
Hi, Satya. So good to see you.
I'm going to piggyback on that question. Not all errors are equal, and not all things have downstream effects. So in the context of the work you're doing with MS, where are those boundaries? Do lesions need to be perfectly defined, or just a general location is okay, as opposed to full segmentation? And I think this speaks to this broader question that Dave brought up about human in the loop curation, is how do we think about the risk of not labeling something as accurately as one might want?
Yeah, so that's a really good question that we are trying to answer right now. We that clinical utility study I mentioned, where we had 3 neuroradiologists rate atrophy and lesion count on 90 patients. We're going to have them do that before and after we run segmentation, after a bunch of manual edits. And then what we're doing is we're sending that to neurologists, and we're asking them what kinds of decisions they're going to make based off of the radiology report. So based off of the kind of traditional standard just text report versus the one that has all this quantitative information. And if we find that neurologists, like, it doesn't matter if it's 1 or 5 new lesions, right? As long as there is a new lesion, maybe that's enough for them to make a decision, then maybe we don't need to, be so specific about there are exactly 8 new lesions and, they're in this region. So I will answer that hopefully pretty soon, but I think that is a very relevant question we're trying to figure out. And how much does atrophy play into a role, right? We've seen so much literature on brain atrophy and MS. Brain atrophy, relates to MS progression, which is this big mystery in the space, but will that information actually be used by clinicians in a way that is meaningful? I don't know.
Is there a correlation between the things we observe and the things the clinicians can actually do something about? If there are 10 lesions versus 3 lesions, maybe that says yes, change a drug or change something. When there's brain atrophy globally or brain atrophy in a particular spot or another, that doesn't really translate into clinician do something different. It just
Yeah, that's interesting to know.
The more targetable or actionable from the clinician's point of view of what they do about the thing that we show them, perhaps plays into that.
To be fair, there is one drug in MS called teriflunomide, which has been shown to reduce brain atrophy in the clinical trial. So maybe it would mean switching the patient to that drug. But I would imagine there are a lot more decision points on whether or not you should take that drug. I don't think atrophy would play the only role, but we shall.
And maybe atrophy wasn't in that case the best example too. So yeah, it's okay. Yeah, but point taken. JB.
Hi, JB.
Hello. Super cool talk. Thanks, Anisha. Is 3 neurologists enough? I mean, what's the variability of the neurologist?
We actually have 3 neuroradiologists and 6 neurologists, and we're always looking for more. And it really depends on what they're looking at, right? So, yeah, there's a lot of variability for some things and very little variability for other things. So if you know any MS neurologists, let me know. We'll take more raters.
No, I think it's it is, and the kind of even those measure of like repetition on the same rater for the same rater aspect. It's unclear. Again, I think the critical thing is what do you do with the information. I mean, that's where you can assess how solid and precise should be the information. That's so it's not like the information itself is obviously what you're doing with it, but I'm always surprised by the all those quality check and all those things with 1 or 2 people that are sometimes neuro radio and sometimes not even neuro radio, and I just don't know how much has been solidified in that space. Anyways, I think it's
in terms of how well they agree with each other.
Yeah. Or with themselves, by the way.
Or with themselves. Yeah, it could use an improvement. Yeah.
And I had a second quick question. I mean, maybe for after. I'd love to hear Nikki's questions. And what a bit of the translational aspect of brain imaging aspect. It sounds like after working a few years in that sphere, do you have any word of wisdom of thought of what is actually likely to translate? I mean, we're us working on reproducibility aspect, we're kind of the skeptic crowd here, and I just want to know general impression of what you think of
I think in MS, I think lesion counts are pretty crucial, but they're not maybe as exciting in the research sphere, right? Because it's a long time and it's like, yeah, of course they have lesions. We're more interested in other, advanced imaging metrics. But I think practically speaking, I think lesion counts are really important. I think brain atrophy, it's being used in pharma right now, but translating this to the real world is really challenging because of, scanner and sequence differences and kind of like the US healthcare system. Like, we need time to get a good quality image, right? And we need to be a little pickier about, maybe rescanning a patient if they've moved too much. And I think the hard part is getting imaging centers to change. Like, what is their incentive to do so, right? And I think we're still trying to figure that out. But I think if they can change and get good quality data, then I think the simple whole brain measurement could be pretty cool. I think it could translate. Yeah.
Thanks so much.
Great. The floor is yours.
Can you guys hear me?
Yeah.
Oh, cool. That's exciting. Yeah, hi. I don't think we have met, but I've known about you from JB for a while, and other people as well.
I hope good things.
Yeah, mostly.
Okay.
No, I'm kidding. Yeah, all great. It's a similar question that I guess JB asked, and what how you responded on the data quality and stuff. And I think it mentioned a GAMless and brain charts type of things as well. I'm curious to know more of exactly how you guys look at that in terms of like side effects and scanner effects type of things.
Yeah,
And
so we do include scanner manufacturer field strength and contrast to noise at the moment.
okay.
This makes our model fit very nicely. So this is just the early stage of this. As we get more data and update the model, I think we can start to include more covariates. But at this point, we just want to know if it would even be useful. And so that clinical utility study I'm talking about, the next stage of that will be kind of including these brain charts and seeing if it even resonates with neurologists at the end of the day, because maybe it's not going to make a difference. So in that case,
No, I guess
yeah.
better question would be, how do you know the ground truth in a sense?
Oh yeah, no,
Because
we don't. I mean, we don't.
Yeah.
We have, the qualitative impressions of brain atrophy from our neuroradiologists. Unfortunately, they don't agree with themselves or each other on it. But it is something that is reported in a lot of radiology reports. So they do look at these, they look at the size of the ventricles, the whole brain, and they do make a statement on atrophy. It's just not very reliable. So maybe with a little bit of this automation, it can at least be more reliable, and then maybe it can become useful. I don't know, but that's what we're going to find out.
Cool. Thanks.
Any other questions?
Any other questions? I know in our pre-chatting you were bringing up the historical tool Brander.
Brander. Yeah.
You still see a vision for that kind of thing in the future?
I see a vision. If we can figure out how to, so Brander, for those of you who don't know, was kind of a joke written for Satya and Dave after one of our hackathons, where I first tried to get them to QC data the traditional way, looking at all the planes of the T1 weighted image, and then having them rate it on a Likert scale. And they said, no, we're not going to do this. And so I, on the plane home, started writing a Tinder app. And I just kind of wanted to make them laugh. But what happened was it went viral on old Twitter. And a bunch of people started, swiping right and left on these images from the Healthy Brain Network. And then from that information, we found that actually the ratings really matched up pretty well. Like the average quality rating of an image matched really well with, kind of this traditional way of doing things. And so we were like, whoa, we can take a total shortcut here by just showing a single slice of the brain and crowdsourcing the rest. And using that data, we were able to train an algorithm to, do pass fail on brain image quality. So that was cool. What was really striking to me was that one of my friends, Greg, he swiped on 800 images in 10 minutes. I was like, wow, that is a lot, right? So imagine doing that for something like lesions, where, you can QC a bunch of potential lesions in 10 minutes. That would be amazing. The issue I'm running into, though, is that there's so many false positives. In order to make sure I capture all the false negatives, I get so many false positives, it becomes a very boring game where you're just swiping left all the time. So we're trying to figure out ways to at least enrich the potential lesion candidates to closer to 50-50, so it's a little more fun. And so we're kind of building this improving the automated method just to a point where humans can then, using their eyeballs and swipes, kind of get us past the finish line. But again, we may not have to be that accurate with the lesions. I would love to. Maybe I'm being OCD picky about wanting everything to be perfect, but yeah, I think that's the future. Hopefully at Octave MRIs, everyone in the company while on their coffee break will be swiping for me. Or while waiting for people to get on Zoom calls, they can just swipe.
Can I ask something in relation to those MRIs and Octave and MS? Is there an effort to create open data sets around MS? Because it seems like that could help everyone.
It would help everybody. A lot of the data that we get are clinical clinical scans, and I think we still need to work out the IRB protocols and all that, and include that in. I think it's a little scary when you have MS, because I don't know, I wouldn't want people to know I had MS, right? So I think there's a lot of fear around, because this is a very like clinical serious disease. So I think there's probably some fear about like security around that, but I don't know if there's a way around it. I'm all for open data,
Could be addressed, I think. So I'll ask the other question in relation.
Yeah.
Given these are clinical scans, would tools like SynthSR help in this setting, or are you using it in these settings?
We are not using it yet. I'm a little scared, and maybe I'm just not educated enough in this, but I wouldn't want to use synthetic data and put it in front of a neuroradiologist, because they're going to be like, what if it missed a tiny little lesion, right? Like that's not great. I think in terms of like training algorithms to maybe improve lesion segmentation, yeah, I'm all for that. That would be great. So I think that'll be a future work. Well, Felix, hi.
Hi, Anisha. Cool talk. Thanks. I have a short question. I'm not aware, I don't do work with MS data at all. So, but really nice, the overview and the insights now. My question would be, sometimes if you have private data, it's one of the ways we're doing is combine it with like data sets that are open. And there are a few clinical data sets that are, I know from Parkinson's and of course Alzheimer's and the rest. Are there for MS as well? And are you using it? And if then, how? Because that's would be another general, I think, application, how to effectively use open data sets with private data to make that useful.
Yeah, so right now, there are a few open MS lesion segmentation data sets through McKay. They're quite small, and the sequences that were used back then aren't necessarily the sequences that I see on a day-to-day basis. I think one of the releases had like 4 different modalities, but like one of those modalities isn't really collected that much anymore. So it can be a little complicated. So we are trying to use what we can. But I think maybe federated learning. I think that's got to be the answer, right?
Yes, and yeah, I mean there got to be data to do anything, right? So, and that's I think then the problem in that sense. Right. Yeah. Thank you, and good luck. Really sounds cool. I'm looking forward what comes out of what I do.
Thanks.
Great. Any more questions out there? If not, I will thank everyone for joining, and thank you again, Anisha. It's very great to have you.